Nivedita is here by taking a new video on the topic regulation of gene expression. So this is a topic from class 12th biology, and it belongs to the chapter number 6, means the second unit of genetics. So first of all, means what we are talking of. Regulation of gene expression means what? See, genes all the time, all the genes will not be in active state. Okay, sometimes their need means they are required at different times and at different intervals of time as well. Okay, this is so means if the gene is active, it is needed that it needs to be made inactive, and the other possibility is that the gene is inactive and we need to the body requires it in the active form. Okay, so this is the means in easy word we can understand ki what is meant by the regulation of gene expression. Means we can define it like this ki it is the controlling the amount and time, okay, amount and time of the formation of gene products, because genes are expressed in the form of some products or in the form of some proteins. Okay, means their production as per the need of the cell. So if we talk of eukaryotic cell, so here the gene expression, it takes place at 4 levels. First level is transcription level, second is procession level. Procession means eukaryote we are talking of, means there the HN mRNA, okay, HN RNA, not mRNA, HN RNA, it requires the processing, maturation, we call it as splicing. Okay, so that level we are talking of. The third one is transport of mRNA from the nucleus to the cytoplasm, and the fourth level is translation level. So these are the 4 levels where gene expression can take place. So let we have example of lac operon. It is a lac operon model. So we will begin with what is operon. Operon is not a thing. Okay, operon is a unit. So the concept, it was proposed by Jacob and Monod in 1969. So operon is a unit of prokaryotic gene expression which includes structural genes, the controlling elements, and regulatory gene products. Okay, so it is a unit which is consisting of few genes. So we will go through what it consists of, means components of an operon. So majorly we will just go in under 5 headings, subheadings. So the first component of operon, lac operon, is structural genes. Structural genes are those parts of DNA which are transcribed into mRNA. Okay, the purpose behind this is protein synthesis. Second component is promoter. And what is promoter? So it is a sequence of DNA where RNA polymerase binds itself and initiates the process of transcription. Okay, this is promoter which will begin the process. Operator means this is the sequence of DNA which is close or adjacent to the promoter, and here a specific repressor molecule binds. So that is the operator. Means we can understand like that ki operator controls the structural genes. Means if the operator is on, then the structural genes will be translated. If operator gene is off, means structural genes will not be decoded. Okay, so this is some regulatory gene or regulator gene. So this is the gene which codes for the repressor molecule, and this is the repressor molecule which gets bind with the operator and turns it off. The fifth component is inducer, and this is the substrate. Okay, this is the substrate, not a gene. This is a substrate that prevents the repressor molecule from binding to the operator gene. So this is called as the inducer. Okay, when inducer will prevent the binding of repressor molecule with the operator, then the system will be on. So just we will go through the topic, the lactose operon. What is it? See, the operon, I already told you, operon is a unit, means it is consisting of some genes. So the lactose operon, when we are saying, means it the lac Z, the lac Y, lac A genes are transcribed from a lac transcription unit under the control of a single promoter. And these genes encode enzymes which are required for the use of lactose as a carbon source. So the lac gene product means lac I gene product, lac repressor. These are expressed from the separate transcription unit uh, upstream. Okay, which is upstream from the operator. Okay, Lecopyron consisting of three structural genes, and the three structural genes are Z, Y and A. Okay, these are the three structural gene. Apart from this, there is operator gene, there is promoter gene and regulatory gene I. Okay, and lactose is a substrate. Already I told you this is acting as a inducer. And three structural genes, they are polycistronic because there are three enzymes. Information is of three enzymes. So this is the diagrammatic representation. Here you can see, okay, promoter site orange color, operator gene that is red in color, and the three structural genes green in color, Z, Y and A. And they code for Z code for galactosidase, Y code for the permease, and A codes for the trans-acetylase. Okay, there is a regulatory gene I. So I is a regulatory gene which will further make the repressor molecule. This is the whole process, whole system, whole unit is shown over here. How the lactose act as an inducer. See, there is a repressor molecule, okay, which is pink in color. That is a repressor molecule produced by the regulatory gene. And when this lac repressor molecule will bind itself with the operator gene, then the system will be turned off. And if we add their lactose, which is an inducer, then what happens? This lactose gets bind with the repressor molecule and just make an inactive molecule, and now the repressor molecule is not able to attach itself with the operator. So this is shown in the diagram. Okay, how this happens. First of all, for the three structural genes, their mRNA is synthesized, and further that mRNA is translated. So I told you what the structural genes are. So three genes, and their role are: gene Z is there which codes for the beta galactosidase, and its role or its function is to break lactose into galactose and glucose. And gene Y is there which codes for permease enzyme, and this increases the permeability of the cell. And third one is gene A which codes for trans-acetylase, and this catalyzes the trans-acetylation of lactose. Okay, so this is the role of structural gene. Now what happens when the lactose is absent? When lactose is absent, the gene I produces repressor, okay, repressor mRNA, and which is translated in the form of repressor molecule, and this molecule or a protein binds to the operator and what happens as a result? It prevents the RNA polymerase to bind it with the operon, and as a result the operon is switched off. And now the question, what happens when lactose is present? So already I explained, if the lactose is there, it acts as an inducer and it binds with the repressor molecule to form inactive repressor, means the repressor molecule fails to bind with the operator gene, and the RNA polymerase now is able to bind itself with the operator and transcript lac mRNA, which is polycistronic and produces all the three types of enzymes. Means in this condition the lac operon is switched